Hello everyone. The topic is structure of immunoglobulin or antibody. So this is the topic of immune system second year zoology. So this is important 4 marks in the IPE. So here we have to know the structure of antibody. So before that, the antibodies are produced from the B cells or lymphocytes. Especially from B cells, we have two types: memory cells, plasma cells. The memory cells will store the information or recording the antigen, whatever enter into our body. Then the plasma cell function is to produce antibody. So here we have to know the structure. The another name of antibody is called as immunoglobulin. So here when the pathogen is enter into our body, then the B lymphocytes produce antibodies. So in that antibodies, specially for the specific antigen. So whatever the antigen is, antigen or pathogen, both are similar, enter into our body. So regarding the same specific antibody will bind to the antigen. Then again the defense mechanism between the antigen antibody, it's showing the antigen antibody reactions. So if the cell will burst and the substances will secrete out from the cell. So this is the antigen antibody reactions. So that much importance of having antibodies in our body. So that will boost the immunity in our body. So the structure is proposed by the person Rodney Porter. He is the person to describe or propose the structure of the antibody or immunoglobulin. So it is Y-shaped structure of antibody and it has a 2 heavy chains and 2 light chains. So totally it has 4 chains, 2 heavy H1 and H2, 2 light L1 and L2. These 2 type of chains bind with the disulfide bonds. So same insulin structure also, the A chain, B chain together combined with the disulfide bond. Same likewise here also the antibodies bind between 2 heavy chains and light chains, that is the bond is disulfide bond. And one more interesting thing here the antibody will bind to the antigen to do the reaction defense mechanism. It has a structure that is paratope. So what is paratope? This is the MCQ for MCET and NEET. It means antigen binding site. So paratope is a antigen binding site, but it is a part of antibody. But one more structure is there in antigen structure, epitope. Epitope is the part of antigen, but here the part of antibody that recognize the antigen. This is the paratope. This is the paratope. So here is the place to fix the antigen. So that is why how can we define the paratope? This is the antigen binding site. It is the part of the antibody that is called paratope. So the different statements, different questions from this topic. What is paratope? What is epitope? So you have to remember paratope is the part of antibody. Epitope is the part of antigen. Paratope is the antigen binding site. Clear? So here antibody is Y-shaped. It has 2 heavy chains, 2 light chains. These 2 are combinedly with the disulfide bond and it has a Fab end and FC end. Fab end means here fragment of antibody. F means fragment, AB means antibody. So here FC, fragment of cell binding. So it is attached to the another cell binding FC. So one end is Fab end, another end is FC. Fab end means fragment of antibody site, then fragment of cell binding site. So this is the structure of antibody. Then Based on the mobility, the antibodies are two types, that is circulating antibody, surface antibody. Mobility means movement. Based on the movement, antibodies two types. Circulating means they are present in the body fluid. So in the body fluid we have a blood, lymph, right? So from this the antibodies are present. So this is circulating antigen. Surface antibody, name itself indicates surface. In the mature B cells, in the memory cells, the antibodies are present. Not in the fluid, it is on the surface of the cells. So based on mobility. So here we having the antibodies are showing motility. Means they are moving in the total blood or lymph or whatever. But here they are fixed at the surface of the cells. If the antigen will come to near this surface, then only it will attack. If not here, the circulating antibody will circulate in the body fluid. Wherever the antigen, it will go and react. So based on mobility, circulating antibody, surface antibody. Circulating antibody in the body fluid like lymph and blood. Surface antibody will present on the cell membrane. Then based on structure, antibodies types. So here 5 type of antibodies are there. D, E, G, A, M. So here in MCET and NEET also MCQs monomeric, dimeric, pentameric. Monomeric DEG. So here a code, not dog, deg, DEG. Monomeric. Dimeric A. Pentameric M. So here just like 1, 2, 3, 4 you have to like a dot 5. So pentameric. So this also 2 marks. Colostrum in the first milk of the mother having the IgA antibodies. So these are the antibodies structure. Based on structure, types. Based on mobility, types. So you have to remember the structure of antibody or immunoglobulin for 4 marks question. These are for MCQs. Thank you. If you like the video, please subscribe and share.